Hello students, our next topic is genetic mapping. Morgan, he was the first scientist to demonstrate linkage between genes in Drosophila. So in our previous topic we have studied about linkage and crossing over, and there we have studied that this Morgan, he was a scientist, he demonstrated this linkage in a fruit fly called as Drosophila. He explained that this phenomena was due to the presence of genes on the same chromosome. So in linkage we have studied this, that the genes that are present on the same chromosome and they lie close to each other, those genes are not separated even after meiosis. Crossing over won't take place in those genes which are lying close to each other on the same chromosome, and thus we call those genes as linked gene. Morgan and co-workers found numerous hereditary characteristics that are associated together in a group, and we call these as linkage groups. For example, the white eye, miniature wing, yellow body color in Drosophila, these all genes are present in the same chromosome, and so we call it as linkage group. Morgan explained the opinion that there was a relationship between the recombination frequency of linked gene and their linear distance along the chromosome. Now we will come to what exactly genetic mapping is. In genetic mapping, we used to determine the distance between the genes and their location in the chromosome. So, at what distance two genes are present, that distance can be determined with the help of genetic mapping. And this opinion was given by Morgan. So, Morgan, he expressed his opinion that there must be some relationship between recombination frequencies of linked genes and their linear distance and the distance along the chromosome. For example, we are taking three genes A, B and C that are located in a linear order, that is in a straight line on a chromosome, and if we know the degree of separation between gene A and B and that of the gene B and C, so if we know the distance between A and B and the distance between B and C, then it should be possible to estimate the distance between A and C. Simple mathematics, right? So we know the distance between A and B, we know the distance between B and C. So we can estimate the distance between A and C. So this distance is calculated based on the recombination frequencies in the segregating progeny and is known as the genetic distance, which is expressed in unit centimorgan. So centimorgan is the unit for this distance, for the genetic map distance, and 1 centimorgan corresponds roughly to 1% recombination frequency between the two genes. So we can we can just get a rough idea that 1 centimorgan is approximately equal to 1% recombination frequency between the two genes. So in this topic you need to learn about what exactly genetic mapping is. That is, we can determine the distance between the two genes, then the unit, that is centimorgan. So the genetic distance, it can be expressed in unit, and the unit is centimorgan. So this is all about genetic mapping.